రాస్తాను కానీ ఏంటంటే ఎక్కడ పోతున్నాయి బయట పంపిస్తాను.
అందరు సైన్స్ అయిపోగానే జిరాక్స్ తీసి
మ్యాపింగ్ ఆఫ్ గ్రీన్ పిక్చర్స్ ఇంకెంత ఎందుకంటే అడుగుతాం ఏమైంది ఒకసారి లేకపోవచ్చు అన్ని వేళలో మనకి ప్రింట్అవుట్ చూపించాను. ಕ್ಲಾರಿಟಿ ಇಲ್ಲ, ನಾನು ತುಂಬಾ ದಿನ ಆಯ್ತಂದ್ರೆ ತೆಗೆದ್ಬಿಡಿ. ಫಸ್ಟ್ ಕ್ಲಾಸ್ ಅಷ್ಟೇ ಅಲ್ವಾ ಮೇಡಮ್?
A very good afternoon to all the participants. So, okay. So, good afternoon. So, I welcome you all to this NPTEL interactive session, for the course on pharmacological and toxicological screening assays. So, I'll be very happy to address any queries which all the participants might have. So, feel free to ask any questions, any queries you might be having regarding this course. And I'm very happy that you all have registered for this course. And I think in the next few weeks, we'll be going through the different lectures which we have floated through NPTEL. So if you have any questions, feel free to ask one by one. I'd be happy to interact with you. You may put up your queries in the chat box also. And from the chat box, I will pick your queries and we'll try to address it. So feel free to put your queries in the chat box.
Hello.
Yes, please.
Good afternoon, sir.
Good afternoon.
Myself, Syed. I'm a BPharm graduate. UG graduate. Like I have some doubts regarding my research work, UG research work.
Regarding?
Like it's related to probiotic and gut-associated lymphoid issues.
I could not understand what you're saying. What doubt?
Like my research work is on probiotic bacteria that can be related with the GAL system, gut-associated lymphoid tissues.
But I think have you registered for this course which we are teaching?
Yes, sir. Yes, sir.
I think that's not the related question. If you look at the table of
No, sir.
content.
I know that I'm still. I haven't elaborated my question. Like I want to know animal models. In which kind of animal model we can study this immunological systems in a very detailed manner.
Okay. So your question is little bit different from what we have floated in this course, pharmacological and toxicological methods, because sometimes it is very difficult to put all the different animal models in a 12-week course. Sometimes you need to float more than one course if you want to cover all the different animal models. And your question is very relevant, which is as far as I understand, it is related to the gut macrobiota, prebiotic, probiotic, how they modulate the gut macrobiota and play a role in the different disease condition, right?
Yes, sir. Yes, sir. Especially in IBD.
IBD, intestinal bowel disease, right?
Yes.
So based on my experience, there are several animal models for that. And one of the animal model we are using in our lab as well, where we are, first producing the germ-free rats by treating them with antibiotics, for alternative, like 4 different doses of antibiotic. And then once the animals are germ-free, then we are inducing the nerve injury, because my lab works on the neuropathic pain. So we have one grant from ICMR, Indian Council of Medical Research, where we are trying to understand the role of gut microbiota, how it contributes to neuropathic pain. So for that,
yes, it's actually related. There is a system, I think so. Gut brain.
Yeah, right. So we are working on DRG, dorsal root ganglia. We are working on a spinal cord, and we are also working on the supraspinal regions like brain, to study what are those modulations which are happening before and after neuropathic pain, and also before and after the treatment with different prebiotic, probiotic, and also fecal macrobiota transplant. So we do have certain interesting findings, which we have already communicated in one journal. So the publication will be out very soon. And we did found that the healthy macrobiota is very helpful in terms of suppressing some of the symptoms, not all symptoms, but some of the symptoms of the neuropathic pain. So what I'm talking about is on the neuropathic pain.
Neuropathic.
At the same time, gut microbiota has a diverse role in not only neuropathic pain or neurological disorders, in a lot of other disorders as well. Gut is known as the second brain, right?
Mhm.
So especially in IBD, a lot of research is going on, and there are very good animal models which are available. And a lot of clinical trials are also going on in the field of gut macrobiota and this intestinal bowel disease. Yeah. But it is not the topic in our discourse. But
Okay.
if you're more
Like I
interested,
just like I thought you would have expertise in this. That's why.
no problem, no problem.
Uh,
Maybe in the next course, which will propose the extension of this course, we will try to cover the other animal models also, including intestinal bowel disease or many other disorders are still left, which we could not cover in this 12-week course.
Sure, sir. Like just one thing. How did you like which antibiotics you used to entirely remove? Like how did you make a rat germ free? Which antibiotic was
So it's a
used and in
standardized
which way?
Yeah, so it's a standardized protocol in which you have to use the mixture of two different antibiotic and you treat the animals, alternative day for 4 times, and it is well reported. So that makes the gut, like germ free basically. So whatever bacteria, whenever we take the antibiotic, we also get the diarrhea, and that is because of the killing of the good macrobiota in our gut. So antibiotics are ultimately responsible for killing the bad macro along with the good macros as well. So that is one of the well-established, way of inducing the germ-free animals, making of the
Okay,
germ-free animals.
okay, sure, sir. That protocol, like, is it available? Where it is
Yeah, it is.
written?
It is already reported in a lot of publications which are related to gut microbiota.
Oh,
And if you're interested, feel free to send me an email. I will share the detailed protocol with you.
sure, sir. Sure, sir. Thanks a lot. Thanks a lot.
So good afternoon, Nagraj. I think Is a- your question is related to new innovative alternative animal screening model. So I'm not very sure like in which reference you're talking about, Nagaraj. If you can specify, if you are here, you please feel free to switch on your audio and we can talk, because you're talking, you're asking about the new innovative alternative animal screening model. But screening model for which particular disease we are talking about? Every other day, lot of research is going on, lot of different animal models and other in vitro ex vivo models are being discovered by the scientists every other day. So it depends, what disease you're talking about. So if you can be
Yes,
more
sir.
specific in your
Yes, sir.
question,
Excuse
I'll be
me,
happy
sir.
to answer it. Nagraj.
Sir, actually, already we were discussed about older models, but so many animal models we were discussed. I'm asking about modified and alternative models are not discussed. So that's
Or
modified and alternative new innovative models are not discussed. So any alternative new models are available, one or two methods that will be useful for us. So that's why I asked to you,
are
sir.
you are you asking as alternative to animal model?
Animal model, already we were discussed older models only. So I'm asking about some chapters, any chapter, I'm asking new methods, any one or two
Yeah,
methods.
there are a lot of research is going on in this area. If you look at the new field related to organoids, now 3D organoid models are available where people are trying to, if you go through our content, where we have discussed the alternative to animal experiments, we spoke about the different what is the need to animal alternative, what are the different in vitro models, what are the different stem cell therapy scan pet model, which are well known and the well established model. But if you look at the recent research which is going on, a lot of work is going on in the field of organoids, cell culture based models, right? And in cell culture also, like dual cell culture, where people are putting cells from two different tissues. Like, for example, if I talk about the diabetes research, right? So people are, putting the different cells, like cells from the intestine and cells from the pancreas. So that because one mediator is released from the intestine, which further stimulate the pancreatic cells to release insulin. So now a lot of modifications are, being done day by day. A lot of innovation is being done day by day. Similarly, in the field of organoids also, the 3D models are available where people are trying to, because the limitation which is associated with the in vitro model is that they are cell-based model, where we are working in isolation with a particular cell, like maybe cell from the pancreas, cell from the brain, cell from the liver, or maybe cell from the kidney.
L6, L7 cell lines like that.
Yes, yes, yes.
L7 cell lines like that.
Yeah, I'm answering
Yeah,
your
okay,
question. You know? So what is happening in cell-based model actually? What are the limitations? The drug, when you are giving in this isolated system, the drug may look potential in terms of, releasing some mediators or producing some beneficial effect and not showing any toxicity. But human biology, the physiology, is very complicated. When a complete organism, not only humans, also animals, when it comes to in vivo system, the biology is very complicated. You take a pill through the oral route, it goes to the stomach, and then it gets absorbed through the stomach, through the blood to different parts of the body. But sometimes you don't know what is happening to the drug in the stomach. It may be labile to the acid which is present in the stomach. There might be certain enzymes which may degrade the drug, which can decrease the potency. So these kind of effects you cannot check in the isolated system. Also, the drug may be biotransformed, through the liver. It may become less potent, less efficacious also, or inefficacious also. So all those possibilities are there, which you cannot study in the isolated system using cell culture based models. That's why in vivo systems are required. So recently, if we talk about the organoid based model, so they are the models which are not complete organism, and they are not only cell based also. They are intermediate to this, where scientists are trying to develop the organs from the particular cells. Like they are trying to develop the model of the liver, model of the kidney, and then studying, the effect of particular drugs, mediators, what is happening. And sometimes they are also able to study the two different, basically, organs together, how they are, interacting and modulating the effect of, particular exogenous compounds. So yes, a lot of research is happening in this area. If you're interested, you can get in touch with us personally and we can discuss more about it.
Okay, sir. Thank you. Nice explanation. Thank you, sir.
Sashi Pratap Singh ji, good afternoon. I want to ask that the final exam of this course will be objective or subjective. So, okay, thank you so much for raising this question, Sashi Pratap ji. So this exam will be objective type questions, MCQs, So you have to prepare accordingly. The next question is from Mayur. So I'm preparing for the GPAT and I don't have time. I managed time to attend the course class. I will continue the course or leave it for GPAT preparation. So I would suggest, you don't leave the course in between. Try to attend as many lectures as you can, because once it comes to the assessment, the questions are based on lectures only. So it's good that you are preparing for the GPAT exam, but you registered for this course. I would strongly recommend not to miss the classes. Any other questions from participants? Feel free to ask. Don't hesitate.
Hello, sir. Syed again.
Yeah, sir. Please ask.
Sir. Like I have since I am just a UG guy. Like I want to know.
Hello.
Like, hello, am I audible?
Yes, now you're audible.
Like since I'm just an UG guy, I want to know, like, is there any like how like places that I can go to as do as an intern or a research scholar, not as a research scholar, like as an intern or somewhere in places where I can like go to do these kinds of work, sir. I need some guidance. That's why I'm asking.
I got your question. So you are still an undergrad student, right?
I have recently completed my UG.
Yeah. So I think this course is very helpful to the undergrad students as well, because once you come to masters, it doesn't matter whether you choose the branch in pharmaceutics or pharmacology or pharmacochemistry or pharmacognosy. The students from all different specialization, they have to learn all different models, because you might be working on some disease condition. You might be synthesizing some molecules for particular disease like cancer, Alzheimer, Parkinson. Or if you belong to like formulation, then also you'll be developing certain formulations for the treatment of certain disease. So whatever is the scenario, you have to basically you have the knowledge of different animal models, how to develop them and how to study them. drugs formulations, by using those models. So, and if I come specifically to your question, how you get the opportunity to work on these models?
Yes, sir.
For that,
Hands-on work.
yeah, no, you can get, because last year only, I think in October 2024, we organized one international conference or neuroscience school, basically, which was funded by International Brain Research Organization, which is an international, a neuroscience school funded by IBRO, where participants from different parts of the India and Asia Pacific region, they came and attended. So there we had a lecture from different professors from India and also from the different countries like Australia, Thailand, Malaysia, and different countries, and they came to IIT BHU and they delivered the lecture. Not only that, in the first half we had the lecture, in the second half we, uh, you know, uh, we gave the opportunity to candidates to perform the hands-on training on different techniques like different surgery in the animals, different molecular biology techniques, and also teaching them different behavior models for the assessment of neuropathic pain. So if you get the opportunity, so I would recommend you to work hard for your GPAT exam and then try to join a good institute for your master program. And once you are in a good institute, I think the facilities will be there where you can use all these models.
Is there any will there any places that provides intern opportunities to work
Sorry?
on these models?
Sorry?
I think someone else's mic is also on turned on.
Someone?
Someone else mic is also turned on. I think.
Yeah, I request everybody to please turn off your mic. Yeah, please turn off your mic. Yeah, please ask.
Like I'm asking I wanna get hands-on experience during this period. Will
Yeah,
there be any institutes that
I think you can contact different labs, in India, where, different, these instruments are available. And then you can, there is internship. If I talk about IIT BHU, in the summer, like from, I think around May, June, July, 3 months, we have internship in our institute. You can contact professor, through email, attaching your CV, and then ask that you want to learn these techniques and want to apply for internship. So it's very straightforward procedure. It's almost free of cost. Just 1,500 rupees is the registration fees. And then you can join the lab, and then you can work in the lab, and then you can learn all the different techniques in the lab.
is it also done in winter as well, sir?
Sorry?
Is it also done in winter as well?
No, in winter we don't have this,
Oh.
2 years back it was like throughout the year, but recently the guideline have been changed. So we just offer it in the summertime only.
Okay, sir.
Yeah. But you can check with other IITs and other institute like NAPAS as well, if they allow you to go for the internship.
Sure, sir. Thanks a lot.
No problem.
Hello, sir.
Yes, Akash. Yeah, please ask.
Good morning, sir.
Good morning.
Sir, I am actually pursuing my B Pharma final year.
Okay.
Basically, I am from Karnataka, sir.
Okay.
I have saw some Stanford online courses on LinkedIn and other social media websites also, sir.
Good.
And the main topic of that courses were the drug discovery based on the pharmacology only, sir.
Okay.
My question is, how can I access to these courses online, and are there any funding agencies which help the students who are interested in such courses? So I want to know how to pursue such courses.
You mean to say the courses which are relevant to the drug discovery of pharmacy, right?
Ha, sir. Mainly, I saw a drug discovery topic from Stanford universities.
Right. So yeah, that's a good question, Akash. So there are a lot of institutes basically, like Harvard, Hopkins, Stanford, and a lot of other courses, institutes, which offer these courses almost free of cost.
Okay,
But there are many courses which are paid as well.
okay,
So
sir.
for that, you have to follow their web page.
Yes.
Or sometimes you have to write an email to the administrator whether these kind of courses are available or not. Or you can also feel free to contact the instructor of the course if they are willing to share the link or the content with you. And as far as give me 1 second.
Okay.
Okay. So if I talk about India, in NPTEL, right,
Okay.
some of the content is already available, which is recorded by professors previously. But
Okay.
regarding pharmacy, now more and more content is being, recorded and uploaded. So recently, one more course has been approved by, NPTEL, thanks to IIT Chennai and the NPTEL for approving that course. So
Okay.
we are floating that course, from the next semester, from the January. And that course is on the general pharmacology. So I'll be taking that course from the
Okay.
next semester. So pharmacy courses are, you know, will be there, very soon.
Okay, sir. Okay.
Some of the
Sir,
content is already there, but more pharmacy courses will be there very soon.
okay. Sir, another questions.
Yeah, please.
Sir, I'm basically targeting after masters. I'm basically targeting for the jobs related to the clinical research, clinical development, and like medical writing, regulatory such jobs. I'm targeting, sir. How should I identify my interest based upon to concentrate on particular jobs?
So,
Means in a bunch of opportunities, which should I choose as per my capabilities, sir?
so you are a BPharm student right now,
Yes,
right?
sir. Currently, final year. In 2 months, I will be completed my graduations.
Okay. And you said you are interested in clinical research, right?
Yes, sir. Yes, sir.
You are in final year right now?
Yes, sir. In 2 months, I will be graduated, sir.
Okay, okay. So I would suggest talk to your seniors,
Okay, sir.
talk to your professors as well,
Okay.
and see your interest as well. What are the things which attract you more? The
Okay.
kind of job, the kind of work profile in which you feel more comfortable, and
Okay.
because there are a lot of opportunities, after pharmacy,
Okay,
but
sir.
that depends upon your interest. I
Okay.
always recommend my student to go for higher education because after BPharm options will be there.
Okay, sir.
Pay scale, the salary will be very less. So if you
Okay.
just want to do the job after undergrad, after BPharm, the salary may not be very attractive. So if you
Okay.
want to attain a good position in your career, then you
Okay.
have to upgrade yourself in terms of qualification. So I would recommend go for at least master degree, and then the opportunities will be more for you.
Okay, sir. Thank you, sir. Thank you so much.
So there is one more question from Kavi Patel. So what kind of exam has been conducted? Type of multiple choice based questions or on theory basis? So Kavi, we already answered this question. So this is a multiple choice question based examination where will be provided with the objective based questions, and then you have to select the right option. I hope I answered your query. So there is a question from Ekta Sirohi. Good afternoon, sir. As we know, animals and humans process chemicals differently because their liver enzymes vary. So, sir, how does this affect the accuracy of predicting human toxicity from animal tests? So that's a very good question, Ekta. So you have asked a very relevant question. So the physiology of humans versus animal is completely different. Give me 1 second. बंद कर दो इसको. Okay, so that's very true. The physiology of animal versus human is not same. They are different from each other, right? But have you heard about the thalidomide disaster? What is thalidomide disaster? So let me tell you a short story so that you can understand. You can understand the basically the relevance of the proper testing before we go for the human trials, before we go for the clinical trials. So thalidomide was a drug which was discovered, in the early 90s in Europe, and that drug was being used for the treatment of basically morning sickness in the pregnant females. But unfortunately, the drug came to the market without, detailed testing on the animal models and other models as well. And the result was that thousands of kids, infants were born all over the Europe without limbs, the disease known as phocomelia. And many children were born with mental retardation, physical retardation. They were born without limbs or sea-like limbs. किसी के हाथ नहीं थे, किसी के पैर नहीं थे. So you can imagine this, the situation, the condition of those parents, and the children. Some of those children are still living, with us. And it is just because of one small mistake. The drug did not screen properly in the proper animal model system. So now you're right that animals and humans, they are different. That is the reason why, out of 10,000 molecules, only few of the molecules they go to clinical trial. Rest of the molecules, they do fail either in the preclinical studies, in the animal testing, in the lower species like rat and mice. And if they pass through that phase, then they go to the higher species like dogs and monkeys. And there some of them get failed. And even if some of the molecules, they pass that phase, they go to the clinical trial. And in the clinical trials, especially in the phase 1, a lot of molecules, they get failed because they do not pass the pharmacokinetic, studies, which are, very much required, for a drug for a drug candidate. So that is the reason why, so many drug fails even before entering the clinical trial and also after getting entered into clinical trial. And that is because of the difference in the physiology of the human versus animals. So yes, we cannot compare between the animals and the humans, but humans are not guinea pigs. So we cannot test any molecule directly in the human beings without ascertaining its safety specifically. So that is the reason, why we always go for the detailed, like in silico, in vitro, and then preclinical studies before we even try to test a single molecule on the humans, especially in the patients also. Okay. So there's one question from Aripaha that I have not completed 2 assignments, whether I'm eligible for exam or not. This query you can write to NPTEL and NPTEL will reply back to you, whether you are eligible for exam or not, if you have not completed the assignment. So please feel free to write to NPTEL through email. Okay. There's one question from Surbhi. Good afternoon, Surbhi. If I plan to conduct an in vivo study on Alzheimer's disease using transgenic mice, it would focus on targeting the drug delivery system. Transgenic mice are being used in BHU. So right now, in our department, in our institute, we don't use the transgenic animals for studying the Alzheimer's disease. But that's not a problem. If somebody want to use the transgenic animals, you can procure it from Charles River's laboratory, or there are other suppliers in the United States or in the Europe, which can, uh, uh, properly, supply you these transgenic animals if you want to use them. But right now we don't use transgenic animal for Alzheimer's disease at IIT BHU. But yes, we do other animal models here for Alzheimer, like we do like model of amnesia, like scopolamine-induced amnesia, or we also use the histotaxic surgery, A beta injection, amyloid beta injection. So we directly inject the amyloid beta into the cortex of the animals and then see how it is modulating the learning and memory of the animals and whether that can be, corrected by giving the drug treatment. So these models are very quite commonly used in our lab at IIT BHU. Any other questions? Feel free to ask. I can see Professor Mahavir Dhobi is also here. Yeah, Professor Mahavir, if you have any question, feel free to ask. Yeah, feel free to ask, if you have any questions, I'd be happy to, address your queries. So there's another question from Surbi. Can outsider use your lab for in vivo study? So I think, as we are in IIT, so I think it is our responsibility to support, the research and the study, from different part of the country. So that depends. You can, approach through your supervisor and you can discuss your study. And if you have those facilities in our institute, we can, talk about it. Yeah, definitely. We'll be happy to help. It depends, if the facility is available in the lab, then there is no charge because no consumables are involved. You're talking about the charges in the study. So of course, if the instrument you are using, the facility you are using, which are in our lab, so there are no charges for that. But if you plan to use the equipment which are in the central instrumentation facility of IIT BHU, like high-end equipment, so which are chargeable to us as well. So for those instruments like using the SEM facility, like scanning electron microscope, TEM, or confocal imaging, or other high-end, equipment. For those, we also have to pay, so it is chargeable for everybody. And if some consumables are required, then that you have to take care. But other things which are available in the lab, they are not chargeable. They're free of cost. There is one question, again from Ekta, like how do researchers choose the most suitable animal species for a particular drug or chemical test? That's a very good question, actually, like how do the scientists, decide that what could be the best animal species for a particular drug or the chemical, right? So generally, as a researcher, whenever we want to study any drug, we initially, if I talk about the modern drug discovery, we have more advanced tools with us, like in terms of, computation techniques, artificial intelligence, machine learning. So where we can actually screen the library of molecules. So even before, talking about the animal model, the first thing which one is supposed to do is to screen the library of molecules, by using these AI/ML tools, and then come up with the most suitable molecules in terms of the hits, or you can also call them leads, having the best efficacy and the least toxicity. And then, take the help from the chemist around you to synthesize those molecules for you. And then you can test those molecules first in the in vitro system, I would suggest. Okay, so go for the cell culture based models because animal life is also precious. And if, for example, if you have to test, if you, for example, out of 10,000 molecule, you have done the screening using maybe Schrodinger or using supercomputer GROMACS, right? All those softwares, you come up with maybe like 20 leads, 20 potential hits. You cannot screen all those 20 or 30 or 50 hits in the different animal models. That will be requiring almost more than thousands of animals. Animal life is also precious. That I already spoke in one of the unit, that is week 2, alternative to animal experiment, like animal life and also ethics in animal research. Animal life is precious, so we cannot just sacrifice their life irrationally without any logic justification, right? So even before we go for the animal testing, the first job is to basically go for the in vitro screening by using cell culture based models, where we can actually identify the potential hits, out of those 20, 30, 50 molecules. And then the top 5 hits, we can go for the animal one. Now the question is how to select the best species. That's the question you have asked, right? So this generally we go by the literature. If somebody has already done the same kind of research in the past, then we can get an idea. Okay, give me 1 second. Let me open the chat box. If somebody has already done that kind of research, in the past, so we can get an idea. Okay. Another thing, mostly we go for the smaller species first, like rat and mice, and then see what is happening in those animal models. And then we can go for the higher species like rabbits or, guinea pigs or maybe other high species. But most of the institute don't have the large animal house facility, so that is done in the industrial setup only. I hope I answered your question. Yeah, please mute yourself. I request everybody to kindly mute. Can I request you to mute? Can you please mute yourself? I request you to. NPTEL team. NPTEL team, can you please mute yourself? Yeah, thank you. Please mute. Okay. Now, I think there are a few more questions. Can you explain the steps regarding amyloid beta generation in mice? So I'm not sure what you exactly want to ask. Whether you are asking about the animal model, how to develop the animal model of A beta induced Alzheimer's disease, that's what you are asking, or you are asking the steps of amyloid beta generation. So amyloid beta is a peptide basically, right? That is continuously, getting formed in the brains of all the species, whether animals or humans, right? And you should know the different enzymes which are involved in the cleavage of different, amyloid beta peptides, right? So amyloid beta gets formed, 40 gets formed, amyloid beta, other fragments gets formed, but only amyloid beta 42 is toxic because it is non-soluble, it is sticky, it cannot get clear out from your system, right? And that is how it leads to the formation of amyloid beta plaques, neurofibrillary tangles, which leads to the disruption in communication between the different neurons. And all the neurons, they depend on each other. Like we are talking to each other, each and every cell of your body, whether it is neuronal or non-neuronal, they talk to each other. When they don't talk to each other, they cannot communicate with each other. Sometimes they start, dying. So that's exactly what happens, when neurofibrillary tangles gets formed, their communication gets disrupted, and there is, generation of pro-inflammatory cytokines, and then apoptosis and all those things gets happened, and then neurons get died over a period of time. If you can be more specific, I would be happy to answer your question precisely. There's another question from Survi. Can explain step. Okay, not this one. So without AI/ML tool, it is not possible. Why not? The AI/ML tool are just the alternative tool to help you to screen down the library of molecules. If you have a better tool where you don't have to sacrifice the animals irrationally, why you want to sacrifice the 10,000 animals? If your job can be done in only using the 250 animals or 100 animals. So there is no need to go for that, right? So we have a lot of databases, right? And AI/ML are very powerful tools these days, right? Like Schrodinger is there, Gromax is there, which can help you to understand the affinity between the specific target. It may be a receptor enzyme and the ligand, the drugs which you are screening, to understand, what is happening basically, whether they are having the best fit or not. And based on that, you can screen the molecules, right? So they are just, supporting tools to help you to screen the molecules. So Nagaraj is asking, please explain toxicological studies, fix the dose of animals and how to convert the animal dose to human for conducting the clinical trial. So this is already, if you go through the OECD guidelines, it is already mentioned in detail in the OECD guidelines. There are fixed formulas, by which you can convert the human dose to animal dose, and even a specific animal dose to human dose, like mice dose to rat dose to rabbit dose to guinea pig dose, and even to the dogs, and vice versa. All those different species dose can be converted to the human dose as well. So these formulas are already well reported and given in different, guidelines, including, if you just go to the different literature, you will find it very easily. So yeah, and these formulas are based on specifically body surface area. So the more the body surface area, the depending on that, dose will get changed. Okay, and that's and as far as identifying the doses, for the toxicological studies are concerned, there are specific guidelines which are given by the in the OECD guideline, different OECD guideline, depending upon the whether you want to go for the acute toxicity study versus subacute toxicity studies or chronic, subchronic toxicity studies. They can range from 14-day toxicity studies to until like 90 days or sometimes 2 years studies as well. So depending upon what your objective is, there are already well-defined guidelines which are mentioned in the OECD guidelines. You can follow them to actually, investigate the toxicity potential of your drug or formulation. So Rishikesh is asking, currently researchers are talking about zebrafish model and Drosophila model. Is it possible in every lab? And please discuss its requirement. Please focus. Okay. So I understand, Rishikesh, you are an M.Pharm student and you might have heard from your seniors, from your teachers regarding the zebrafish model and Drosophila model. Look, there are different species, which different labs are using actually to study the effect of different drugs on different disease condition, right? Now, depending upon the feasibility and availability of the animal models in the lab, people are trying to use those species. But sometimes the labs which are very well equipped, which are having the good infrastructure, they do have all the different species. Means they are having zebrafish facility as well. They have Drosophila facility as well. If I talk about BHU, IIT BHU, we do have the zebrafish facility in our department. Drosophila facility is also available in the Department of Geology, which is very close to our department only. We have the rat and mice facility. Even we have the approval for the rabbit and guinea pigs as well. So all these different species, we can study in our department. So the thing is that which species to go for. Again, as I mentioned, you have to go through the literature. What question you want to answer, that depends on that. Definitely, larger species are always better. But it is always recommended, you should screen your molecules first in the in vitro models. And after the in vitro model, you go to the in vivo models. I personally feel that, if you want to answer all the questions in the zebrafish and then you want to extrapolate your finding to the humans, that may not be highly relevant because zebrafish is very distant from humans, right? And you can say the rats and the mice, they are little closer to humans as compared to zebrafish because they have the well-developed organs just like humans. They have a four-chambered heart, they have kidneys, they have lungs, they have ovaries, they have testes, right? Which you can compare with the humans. They have a whole metabolic system like humans. So that model is more closer to the humans. So if you have both the facility in your department institute, I would recommend to go for the smaller animals like rat and mice. And then definitely if you go to higher species like rabbits and monkeys, and mammals, they're much closer to the human as compared to the rat and mice. But it is not easy to work on the larger animal models because those facilities are not available in the academic setup most of the time. So for that, we need to have the industrial collaboration. But sometimes these smaller species are useful because they are easy to manipulate genetically, especially Drosophila and zebrafish-like model. So these models, they have certain advantages over the other model. If you want to do the genetic manipulation in the rat and mice, that might take more times. Okay. And it's sometimes easy, and sometimes, specific kind of studies, like zebrafish are transparent. That is another advantage with them, right? So I would not say that any one species is, like the best model, right? You need to understand what are your objective, what questions you want to answer. Depending upon that, you can go for the right animal model. Okay. I hope I have answered your question. So what cell culture models will be asking, what cell culture model can we use in the Alzheimer's disease for targeting drug delivery system? There are a lot of models for the cell culture based assays, like you can use the neuronal cell line where you can, try to culture the neuronal cells, especially from the cortical and the hippocampal region, because cortex and the hippocampus, these are the two brain regions which are involved in learning and memory. And then you can induce the neurotoxicity by using the A beta, amyloid beta, which is available commercially. So you just read the literature, articles, the specific concentration you can put in these cell lines, neuronal cell lines, and then see the toxicity. And at the same time, the other set of cells you treat with the drugs, and then see whether A beta induced neurotoxicity can be inhibited in those cell lines. Okay, and that can easily be done by using the simply MTT assay or other apoptotic cell-based assays. So there are a lot of literature on that. You just have to read the articles related to that. So Anurag is asking, I'm finding difficulty in identifying topic for research in MPharm. So, so I think you should talk to your supervisor, whoever is your supervisor, and of course I would be happy to help you out, but it depends upon the institute, the department where you're working, what kind of facilities are available there, right? So if I would recommend you to work on certain area, and if those facilities are not available in your institute, it is of no use. So I would recommend you to please talk to your supervisor and from supervisor and ask him or her to help you to select your MPharm project. So Niranjan Behera is asking if you can share some insights on cell culture model, cell lines for reported toxicity studies. Yeah, of course, there are several, cell culture based models, for the reported studies as well. And we have discussed, some of those models in one of our, unit as well. I think that is in the, yeah, so I'm just trying to find out which week we have covered that. So that is in the week 11. If you go to the week 11 of our course, which is about the other toxicity studies. So there we have the first unit which we have discussed is on the reproductive toxicities. So there we have discussed some of these models in detail. But if you are interested, we can talk more about it. Feel free to reach out to me through email or phone. There are a lot of models on reproductive toxicity. Parveen is okay. So Parveen is asking, can we connect to you over WhatsApp for doubt? So I don't mind if you connect to me through WhatsApp, but I would suggest, you please write me email, because sometime due to busy schedule, I may not be able to, respond on WhatsApp very frequently. So if you write to me through email, I would try to get back to you as soon as possible. And the next question is how to get the ethical approval for animal testing for fungus. So I think your institute, if they have the institute biosafety committee, and if your fungus is not infectious, so again, it depends whether you want to study the effect of fungus on certain animal model, then you need two different kind of approval. First approval which is required is the institute biosafety committee approval, because you are using microorganism, whether they are pathogenic or not, that doesn't matter. But for that, you have to go for the institute biosafety committee approval first. And based on that certificate, you can approach your institutional animal ethics committee to get the another approval. And that's very easy and straightforward, way to get the approval. Another question is, can we do organ chip model for Alzheimer's disease? Which organ chip model are available within the budget? So there are several organ chip models for different disease. I'm not exactly, aware of the costing of this kind of, devices. But yeah, people are, developing these models in their lab who are working in this area. I especially do not work on this area. So I may not be the best person to tell you about the costing and availability of this model. I'm putting my email ID here, and feel free to reach out to me for any queries you have. vtiwari.phe@iitbhe.ac.in. There's another question from Ekta. In acute toxicity studies, how is the choice made between mice and rat? So you just go through the OECD guideline, Ekta. So these are the guidelines which are not even followed by India, but also by the other member countries who are member of OECD. So it is very clearly mentioned, like what species you should mainly go for. As far as I understand, they clearly mentioned rats in their guidelines. So
Sir, you're on mute.
So Mrs. P.S. Bhandari is asking, what is the best model for the ulcerative colitis? It is very difficult to say which is the best model because, again, as I mentioned before, it depends upon the question which you want to ask. Based on that, you have different models actually. Some models help you to investigate the pathology of the disease in detail, right? Some model help you to screen the molecules, in terms of the symptoms which are associated with the disease. And some models are there which can serve both the purpose. So depending upon the question you are asking, the model, can be, good or bad, basically. So I would not recommend any one model as the best model for any disease, whether it is ulcerative colitis or any other disease. But yes, depending on the question you want to ask, that's why the scientists, they always go for at least more than one models and at least more than one species when they want to claim that their drug is efficacious, because it is quite possible that your molecule may work wonderfully in one model, but it can fail miserably in the other animal model. So it is always good to screen your molecule in different animal models to uh, test its efficacy and potency, among diverse animal models of that disease. Also, the best model for the any disease is the human only, right? But we cannot go for that directly without ascertaining the safety and efficacy of the molecules in the preclinical models. Any other questions? I'm not sure whether you are getting my trying to put my email ID here. I don't know why it is not. Let me put it to everyone. Okay, now I think it will come. I hope now my email ID is there, right? Yeah. Okay. Yeah, I put it in the chat box now. You can note it down. Any other queries? Any other doubt if you have? No problem. If in case you have more doubts, feel free to reach out to me through my email ID, and I will try my best to address any of the queries you might have. And my best wishes to all of you for this NPTEL course and for the examination. And very soon we will be coming up with the next interesting course on general pharmacology, where we'll be talking on the different, like we studied in B form, right? Like different autonomic nervous system, central nervous system, and then the different drugs, their pharmacology, in detail pharmacology, drug receptor interaction, all those things. So wish you all the best and be in touch. Thank you so much.
Thank you, sir. Sir?
Yes, please. Any question?
Sir, they have NPTEL workshop. No, sir. Like, do you also like plan on doing that? Like
NPTEL workshop,
hands-on workshop, yeah.
hands-on workshop.
Hmm.
So I have not thought about it. I'm not aware about this, Can you
They
send me
do
the
have they have already conducted this in ISR Bhopal. Many institutes around in the IIT Guwahati and many areas. Like, you have any plans on doing that? Like two
NPTEL
yeah, NPTEL workshop.
workshop? So is it like a hands-on training kind of thing
Yes,
to?
sir. Yes, sir. We in person we go there. I actually went there for immunology workshop.
Yeah,
ISR Bhopal.
I think that's a great idea, because many of the students are having the same query today, they
Yes, sir.
want to get the experience of
Hands-on experience.
hands-on using these animal models, which we are already using at IIT BHU. So I think that's a great idea. So you can guide me on how to, proceed for that.
No,
And
sir. I don't have any idea, but I know this thing is happening. I think you can like. I don't have the power to ask NPTEL, I think, but you can. I think you can ask NPTEL coordinators.
okay, so let me talk to, the NPTEL team. Maybe they can help me
Sure.
to understand, figure out, how to set up this NPTEL workshop thing, and I will be
Because if this happens,
happy.
it will be beneficial for all of us.
Yeah, because I know many students, all over the country, they may not have those kind of facilities, which we have in our institute.
Yes,
And the exposure is very important for them.
yes.
So I would be happy to, coordinate that kind of thing. Yeah.
So that I'll be in touch to like how has it progressed to get any information about that.
Sure, sure, sure.
Thank you.
Thank you. Thank you.
Thanks a lot for this.
Okay, thank you. My pleasure. My
When
pleasure.
will be the next session, sir?
I think
Because the first session wasn't conducted, I think. First session like it got cancelled.
sorry,
The first session got cancelled, I think.
the first one got cancelled, right?
Yeah.
I think this is the one, which we have, planned for that cancelled one.
Okay. When will be the next one?
I don't know. I need to check with the NPTEL team.
Sure, sure. Sorry for like barging these questions again and again.
Yeah. Right. Thank you. Thank you so much. So may I leave now? Yeah. And, yeah, feel free to get in touch if you have any questions through email. Yeah. Thank you.
Hello.